Hello.
Yes, sir, you can continue.
Yeah, is it
Yes,
visible?
sir. We are audible and we are visible. Your slide. Yes, sir.
Okay, good, good. Okay. Yeah, good morning again, one and all. So, I think now there was a bit technical issue, so I think they have solved it very quickly. So, thank you, Santosh and Pramod. So, let me begin with my slide. Okay, so my topic is because I was asked asked to give a seminar under a heading called Immunity and Infection Biological Approaches for Therapies. So what I thought that let me talk something about very basic, that very important. So that's why I have chosen the topic, that is MHC molecules, central regulators of adaptive immunity. So as you see the topic, MHC molecule, central regulators of adaptive immunity. So the thing is, I have divided this topic into three parts, that is MSC molecules and adaptive immunity, and how these two aspects are connected. Okay. So if you see this, before going into the details, let me talk something little bit about the immunity. So the immunity, so as we know, the immunity is nothing but it is the defense mechanism against different pathogens, right? So in short, the immunity is nothing but the protection from different diseases, right? So the immunity could be two types, as we already know, and we have studied it from many days, many years. So I want to give a brief introduction. Briefly try to discuss about those different immunity. So the first thing is innate immunity. So as we all know that innate immunity is the non-specific, which is also the first line of defense against foreign pathogens, and which is an integral facet of the immune response. And the major component of this innate immunity is like different type of cells, particularly the dendritic cells, macrophages, neutrophils, basophils, eosinophils, mast cells, etc., etc. And apart from these different cells, as you can see, there are many molecules also play an important role in innate immunity. Not only the dendritic cells or the macrophages or other cells, including there are many molecules, say for example, complement system, it could be the inflammatory system, or it could be the other molecules. So they can also play a very important role. But the cell-wise, the dendritic cell, macrophages, and these are the myeloid progenitor. They are originated from the myeloid progenitor cells. So they play an important role in executing innate immunity. And next, that is the adaptive immunity. So the adaptive immunity, as we all know, that it is a highly specific second line. In fact, some people, they call it as a third line of defense also. So the definition is like the non-specific, the specific second line of defense against foreign pathogens produced in response to particular type of antigen, which is very important. So it is a particular type of antigen or pathogen. So that is how the adaptive immunity differentiate from innate immunity. So here, innate immunity is a kind of non-specific and which is very quick, although, but the adaptive immunity is a kind of a very specific and which is bit slower side. So it executes, it takes some time to build the adaptive immunity and then it executes the immunity. So the major components of adaptive immunity are B lymphocytes, T lymphocytes, NK cells, APCs, that is antigen presenting cells, as well as all the nucleated cells. So with this brief background, so let me talk about, before going into that, even the adaptive immunity is of, like we can have an active adaptive immunity as well as passive adaptive immunity. So active is nothing but, as we know, it is inherently present in the sense it's produced by the human body. whereas the passive is kind of a ready-made thing that are given from the outside to the body so that to combat any disease or any pathogens. So let me discuss about some major mechanism of innate immunity before talking about the adaptive immunity. So as you can see, the innate immunity is a complicated pathway. It involves many molecules, as I explained already. And the different mechanisms of innate immunity are the physical barrier. So the physical barriers like skin, it could be mucosa, it could be different physical barriers. So what they do, they don't allow the pathogens or any more foreign invaders to enter into the the body. So that is the first line of defense. And this is the first mechanism of innate immunity wherein they don't allow the other molecules to enter into the system. And once they enter by chance, then the inflammation, immediately the tissue cells will start producing inflammatory responses so that they can recruit different type of cells, including white cells or other molecules, so that the immediately inflammation can cause, and then that will aggravate the different innate immunity cells as well as adaptive immunity cells, and then they try to remove those pathogens or bacteria. But if that is not possible, then the phagocytes come into the picture, like mast cells. In fact, primarily the phagocytes are like dendritic cells and macrophages and polymononuclear cells. They all come into the picture, and then they try to remove the pathogens. They try to kill those pathogens, right? And then the very important system, that is the complement system, that will also come into the picture. And then the complement system is the cascade pathway. There are different type of complement system, like alternative pathway and the classical pathway. So I'll not go into detail of those complement system, but as a whole, the complement system, that is a part of this innate immunity, and it will try to eliminate pathogens by making a force or a performance in those different things, so that they can take out or they can degrade any pathogens. And apart from these four important mechanisms, so the many cells, the innate immunity cells, they can also produce different antibacterial substances like lysozymes and defenses. They can also act on bacteria and like bacteria or like the different pathogens. And apart from antibacterial system, the substance, the innate immunity cells can also produce antiviral molecules like interferons and different molecules, so that they can go and they can take care of the viral molecules or viral substances. So this is all about the innate immunity and different mechanisms that are working under innate immunity. So once the innate immunity, once it tries to take over the pathogens that are present or the molecules or the toxins that are present, and in case if it fails or if simultaneously and parallelly, it also activates the adaptive immunity, right? So the once the innate immunity, it sense that, okay, I may not be able to compete completely. So the simultaneously and also parallelly, it activates the adaptive immunity. So the adaptive immunity is activated even through the pathogen themselves, okay? So the pathogen themselves, they can also activate adaptive immunity. And meanwhile, the innate immunity is working, the adaptive immunity starts working, and it starts taking care of the pathogens that have entered and the pathogens that have escaped from the innate immunity. So basically, the adaptive immunity is activated by innate immunity through a different type of cells like APCs, antigen presenting cells, tissue cells, like the cells which are already inflamed, and even the disease cells, the cells which are already infected with virus, bacteria, etc., and such as. So the innate immunity activates adaptive immunity through these APC or tissue cells and disease cells, etc., etc. Whereas the pathogens, they cannot directly activate the adaptive immunity, but they can activate through different cells, and more importantly, they can activate through B cells. Right? So let us see in next slides. I will show how these are executed. So let us discuss about most important mechanism of adaptive immunity, right? So the most important mechanism of adaptive immunity are three types. That is, first thing is humoral, or also known as antibody-mediated immunity, and the second one is a cell-mediated immunity, and the third one is NK cell-mediated or natural killer cells mediated. So let me start with the humoral immunity, antibody-mediated immunity. Once the antigen enters into the system, as long as the innate immune cells, they try to kill those antigens or they try to take over those antigens. Meanwhile, some antigens, they escape, and when they come in contact with, excuse me, so when they come in contact with B cells, right, particularly B2 cells, and when they come in contact with B2 cells, so the B2 cells, they bind these antigens and they become activated, new B cells become activated and they get differentiated into memory cell as well as the plasma cells, or they're also known as effector cells. So once the beta cell, I mean the B cell become active, then it get differentiated into memory cell and the plasma cell. So the plasma cells, now they start producing antibodies. So these antibodies, initially they produce IgM class of antibody, and these IgM class of antibody, they can try to take over the antigens that are present. As you can see here, the number of antibodies that are produced by the B cells as a result of humoral immunity are very, very less. Okay, and also, as I mentioned, these in the pink antibodies and the antigens are red. So what I try to explain, what I try to impart is that like these antibodies are kind of non-specific. Okay, so these antibodies are non-specific in the sense they, yes, they can go and bind, but not exactly where they wanted to bind. So they are produced in response to presence of particular antigen, and this antibody, it can go bind to the particular antigen, or even it can go and bind to other antigens or similar type of antigens. So that's why the initial humoral immunity is a kind of non-specific. So the system is very clever. What it wants to do is it wants to produce a very specific and highly specific antibodies, and also the more number of antibodies. As you can see, we can count here, say for example, there are 20 antigens are present, and the initial, as an initial means, they can only produce 4 to 5 antibodies. I'm just giving an example. So that these, whatever the antibodies that are produced may not be sufficient to take over and kill all the antigens that are present in the system. So what these cells, they cleverly do, while producing these antibodies, they get activated and they take these antigen inside. The antigen that I bind to the antibody, specific antibody, the beta cell and the B cells will take those antigens inside and they chop or they cleave those antigens into small pieces, right? So once they cleave those small pieces and the antigen present inside, then it cleaves into small pieces, and then those antigens are expressed on the B cells, that is B lymphocytes. And these B lymphocytes, now they are recognized by Th cells. So Th cells, if you remember, they are the progenitor cell, I mean the lymphoid progenitor cells, and Th cells is nothing but the T helper cells. So once the T helper cells binds to these lymphocytes, which are bound with the antigen, and the class switching occurs. So what is class switching is here the antibodies like this cell, imagine this cell is get activated into plant memory and effector cell. So these effector cells, they are producing antibodies, a kind of IgM antibody, not kind of, they are producing IgM antibody. But the moment it takes the antigens and then when it comes in contact with the Th cell, it starts switching its antibody production, and this process is called class switching. So now the activated cells, when it comes in contact with Th cells, the B cells that has been activated and that has been switched to produce different type of antibody, it gets differentiated into again the memory cell as well as the plasma cell or the effector cell. So now these effector cells, what they do, they produce a high number of antibodies as well as the different class, that is IgG class. So the comparatively IgG class are highly specific when compared to IgM. So the whatever the antibody they are released, IgG class, they go and they bind to antibody. So there are different mechanism of antibody neutralizing antigens. So let me not go in detail of those. So particularly like neutralization or ADCC, antigen coated or opsonization, like that, there are different mechanisms. But what I'm trying to explain is once the humoral immunity is executed, initially it produces IgM antibody, which are kind of less or not sufficient, and also they are non-specific, right? And once they, so when the immune system senses this, and it immediately takes those antigens, and then it expresses those antigens on the B cells, and then that activated or expressed B cells are recognized by T cells, and then T cells activates these B cells, and then B cells undergo class switching, and then producing different type of memory cells as well as effector cells. So these effector cells, they produce antibodies, and then antibody, they neutralize the antigens. And if you see this, in fact, the antibodies that produced by these cells are highly specific. And let me highlight this portion. So as you can see, the interaction between B cell and a T cell. So this is the same slide. So I will just zoom it. So if you see the interaction, the B cells, as I already explained, they take those antigens, they chop those antigens, and they present those antigens along with a very specific molecule called MSC2, right? So MSC2 molecules are expressed by these B cells along with the peptides that is obtained from the cleavage or breakdown of those antigens that have already entered into the cell. And then the expressed MSC molecule along with the peptides are recognized by TCR of a T cell. So TCR is nothing but T cell receptors. And then these T cells, they recognize these MSC molecule through CD4 molecules, because the T cells are decorated with CD4 molecules. So CD4 molecule recognize MSC, MSC2, and then so the MSC2 molecules, they bind to TCR, so that the T cells, okay, so the T cells, it becomes activated. So once the T cell becomes activated, right, it releases cytokines. So the cytokines could be autocrine function or paracrine and even in endocrine function. So the autocrine function cytokines, what they do, they themselves, I mean, they activate more these T cells, and also because of that, the T cells also releases some other cytokines. So these cytokines now, they go and they bind to different cells, like all the B cells, or they could be in different cells. So once the cytokine they bind, and all these cells which are trying to produce IgM class, they become class is switched, and they start producing the IgG class of antibodies. And apart from this, even the professional antigen presenting cells like dendritic cells, macrophages, and as well as the non-professional antigen presenting cells, they can also take up different antigens as a result of innate immunity. And also, not only they kill those antigens, and they also take those antigens and they present those antigens using the same MSC2 molecule. So that the same immunosuppressive molecules are also recognized by the T cells, and the T cells again they get activated so that they can release those similar kind of cytokines. So now these cytokines, they can perform different functions. So they can go and they can bind to B cells, or they can go and bind to the macrophages, or they can, and even the dendritic cells. Even they can go and bind to different granulocytes, as well as that they can also go and bind to the TC cells. TC cells are nothing but the T cytotoxic cells. So once the cytokines, they go and bind and they activate these different sort of cells, so that the different cells, they can execute immune response. Say for example, the B cells, as I explained, they start producing IgG class of antibody, whereas the macrophages and dendritic cells, they become activated and they can acquire the phagocytic property, and they can grab all the antigens or the pathogens that are present. And meanwhile, the mast cells like the granulocytes, neutrophils, and as well as different granulocytes, basophil, etc. So they can degranulate and release pharmacologically important molecules into the system, and then they can kill those pathogens or the antigens. And very importantly, the cytokines that are released by Th cells, they can also go and bind to the CTL, the TC cells. So TC cells are nothing but the cytotoxic cells. So once the cytokines they bind and they activate, the cytotoxic cells become CTLS, that is cytotoxic T lymphocytes, and then they go and they kill the different cells. So what I'm trying to emphasize here, all these activation is mainly because of MSC class 2. So the MSC class 2 molecules play a vital role or a crucial role in activating TS cells. So if you go back and if you see, if there is no MSC2, whether it is from the B cell or whether it is from professional antigen presenting cells or the macrophages or other cells, if there is no MSC2, so obviously there is no activation of Th cells. If there is no activation of Th cells, so obviously there is no production of cytokines and there is no production of any, these cells will not be activated. So what I'm trying to emphasize, or what I'm to explain, or what I'm to important to say that the MSC2 molecules are very, very important and they play a crucial role in activating Th cells. So this is one part. So let me take the second mechanism of adaptive immunity, that is cell-mediated mechanism. We have already know like what's going on or what will go into this cell-mediated immunity. But the thing is, yeah, there was a message. Okay. So the second mechanism in adaptive immunity is cell-mediated immunity. So what is cell-mediated immunity? Is nothing but it involves the cells. So let us say all nucleated cells, you take any nucleated cells except RBC. You take any nucleated cells, they can say for example B cell or macrophages or granulocytes or any epithelial cells and even the different cells. So once these cells are infected, they become disease or become infected with virus, pathogen, or even sometimes they become disease, like the normal cell become cancerous and also age. What they do, they express another type of MSC molecule called MSC class 1 molecule. Earlier, what we have seen is MSC class 2 molecules. So now these nucleated cells, what they do, they express MSC class 1 molecule. Along with the peptides are a part of antigen from different, either it could be from virus, bacteria, or by other means. So once they express this MSC1 molecule, they are target for TC cells because TC cells contain CD8 markers or a CD8 receptor. So CD8 receptor recognizes MSC1 molecule. And meanwhile, the TCR, the T cell receptors, also binds to the MHC molecule, and then TC cells become activated. Once the interaction occurs between nucleated cells that have expressed the MHC molecule and TC cell, the TC cells activated. So once the TC cell activated, it gets differentiated into memory as well as the effector cell. So these effector cells are nothing but CTLS. So CTLS is nothing but cytotoxic T lymphocytes. So the CTLs are, once the CTLs are produced, they have a capacity to produce a very important molecule called death ligands. So once the TC cell activated, it differentiates into M, that is memory, as well as effector cells. And effector cells have a capacity to produce a death ligands. Okay, so what death ligands they do, they send a signal to all these different cells that have been already infected with virus, bacteria, pathogens, or they have become aged because they cannot undergo lysis. So for example, if the B cell is infected with virus or the macrophage is infected with virus, and assume if they are infected with virus and virus multiply in the cell, and if the cell burst out, so obviously the other healthy cells will get contaminated. So in order to minimize this complication, what these cells, what they do, they take this antigen and they present these antigens along with the MSC1 molecule so that these cells get a signal for apoptosis. So as we know, apoptosis is a superior way of cells going under death. I mean, they wanted to, if they wanted to go uh, I mean, if the cell want to die, they cannot die with a necrosis, because if they die with a necrosis or other kind of cell death, so they can infect the other cells. So that's why these cells, the cells which are infected, they always try to go under apoptosis. But their intrinsic pathway is already blocked, or they may not execute intrinsic pathway because of the infection with virus or bacteria or some other problem. So what they do, they try to send a signal and they try to get the signal from outside so that they can execute extrinsic pathway of processes. So that is mainly through this pathway. That means they get a signal from death ligands. So these death ligands are produced by CTLS or cytotoxic lead lymphocytes. So these CTLS, not only they release these cytokines, they can, I mean the death ligands, they can also produce, the death ligands can also be the part of the membrane, or membrane bound death ligands can also possible. So now these death ligands, they go and they bind to these different cells and they uh, give the signal to the different cells to undergo apoptosis. So not only the death ligands, even the CTLS can also directly go and bind to these cells which are expressing death ligand receptors, and then they can directly kill the cells. So that's why the name cell-mediated immunity. So here the main cells are the CTLS. But now here, as you can see, the main important stuff that is mediating this cell immunity, cell-mediated immunity, is the MSC1 molecule, right? So MSC1 molecules, as I explained, they play now in, I mean, the MSC1 molecule play a crucial role in activating TC cells. Earlier, the MSC2 or MSC class 2 molecules, they have produced, or they played a very important role in executing humoral immunity as well as the other immunity. But the MSC class 1 molecule, they played a very important role in executing or in bringing about this cell-mediated immunity. So apart from these two, that is humoral immunity and cell-mediated immunity, there is another, which is a kind of in between innate immunity as well as the adaptive immunity. But I would like to consider it is in adaptive immunity, but the NK cells mediated immunity. So let us take an example, the normal cells. When the normal cells are there, so the NK cells, they also will be patterning and they will be trying to, I think this, okay, yeah. So the NK cells, so what they do, they express two kind of receptors on their cell surface, KAR and KAR. The KAR is nothing but killer activating receptors, and KIR nothing but killer inhibitory receptors. So these NK cells, they will be patterning and they will be looking for the cells which need to be killed or which are diseased or something. There is a problem with cells are problem. So as part of their patrolling, they always try to come and look for the activators that are present on the cell. So once the normal cells, if the cells are normal, what they do, they also express MSC class 1 and class 2 molecules, so that once the activator, they bind to the KR or the NK cell, they recognize the cell. If it is a normal cell, they also express the inhibitory molecules, that is MSC1 or MSC2 molecule. So these MSC1 and MSC2 molecules, they give the signal to KIR, that is killer inhibitory molecule, so that the NK cell, though it recognizes activating molecule, but it will not kill the normal cells, because the NK cell will recognize MSC molecule, MSC1 and MSC2 molecule as a self molecule, so that the NK cell will not attack the normal cells. Whereas in case of, imagine if the cell becomes diseased due to infection with virus, bacteria, or maybe if the cell becomes cancerous. So in those conditions, what these virus or bacteria cleverly they do, they want to proliferate inside the cell so that they don't want to be killed by apoptosis or etc. So what they do cleverly, once they infect the normal cell, they stop production of these or they inhibit the production of or expression of these MSC2 molecules, right? So that the cytotoxic cells or the Th cells, they should not come and bind to these cells, and the cells will not get the signal for apoptosis or some other immune thing. So that what these cleverly virus do, they stop expressing MS2 molecule. Whereas the cell is also very, very intelligent. What they do, like NK cell, now the NK cell, they come and the pathology, they come and bind to the activators. But due to the absence of the inhibitory signal, now the NK cell will start producing the granzymes. So if you remember, the granzymes are a kind of proteases, serine proteases, and also different type of proteases, and also the perforins, the co-reducing molecules. So these granzymes are released, and then the granzymes, they go and they attack the disease cell and killing the disease cells, which are already infected with virus, bacteria, and cancer. So what is important here is the MSC molecule again. Though they are not present, but what they do without expressing, like if the MSC molecules are not expressed, then they are the target for NK cells. What I'm trying to explain is the MSC molecules, again, they play a crucial role in activating the NK cells. If the MSC molecules are here, right, the MSC molecules are here, the NK cells won't come and they won't attack the disease cells or viral infected cells or any cell. If the MSC molecules are not there, then NK cell will come and produce those granzymes or porins or different molecules, and then the cell will undergo apoptosis or necrosis or whatever by. So the thing is, MSC, they also play here a crucial role in activating NK cells. In fact, the NK cells, they are already activated. That's why they are called NK cells, the natural killer cells. They don't need to be activated. They are always activated, and they always ready to kill any cells that activate, that present the activator molecules. And if the inhibiting molecules are there, then NK cell won't touch. If the inhibiting molecules are not there in the form of MHC1 or MHC2, then NK cell will attack these viral infected cells or any disease cells or a cancer cell, etc. So what I'm trying to explain is that MSC molecules, again, so they play a central role or a very important role in executing humoral immunity, in executing cell-mediated immunity, as well as to some extent in executing natural killer self-mediated immunity, right? Yeah. So apart from playing role in executing different type of those humoral cell-mediated or NK cells, I mean, as a whole, the adaptive immunity, they have a role. They also play an important role in tissue transplantation, as well as autoimmunity and immunological tolerance. So what are these MSC molecules then? So we need to know that what are these MSC molecules. So the MSC molecules, nothing but they are membrane-bound glycoproteins, right? Expressed by all kind of nucleated cells as well as AP cells, that is professional antigen-presenting cells, antigen-presenting cells, and so these MSC molecules are encoded by genes, cluster of genes, in fact, present on chromosome 6, term, in the short arm of a chromosome 6 in case of humans, and 17 in case of mice. So the chromosome 6 in humans and chromosome 17 in mice, they encode for these genes. So you can see, so this is the chromosome 6 and chromosome 7, part of a chromosome 6 and 17. And they are divided into different regions, H1, H2, H3, H, etc., etc. So these MSC molecules were first recognized in 1937 as a barrier to transplantation in mice, right? So probably, I mean, more importantly, the Groger, and Snell, they discovered this MSC molecule while studying blood group antigens. So in mice, these MSC molecule are the region, the genes that code for MSC molecules are termed as H2 region, whereas in human it is called HLA region or HLA antigens. So HLA is because they are expressed on the leukocytes. That's why they are called HLA. HLA means human leukocytes antigens. So they are present on the, initially they are discovered on the white blood cells. That's why the name HLA, human leukocyte antigens. So this MHA region, it almost accommodate 200 different genes. So the region is again divided into three parts, that is MSC class 1, MSC class 2 region, and MSC class 3 region. So MSC class 1 and class 2 are a kind of classic molecules, whereas MSC class 3 are non-classic molecules. Right? So MSC class 1 and class 2, these are the membrane bound molecules, whereas MSC class 3 are a kind of secretory molecules. They usually accommodate different type of cytokines like TNF alpha, TNF beta, lymphotoxin alpha, etc., etc. And to some extent, some complementary complement molecules of complementary, the complement system molecules. So if you see the structure of MSC class 1 and class 2, they almost superimposable. In the sense, they exhibit a very similarity in their three-dimensional structure. But they have very important different steps. So let me talk about the MSC class 1 molecule. So the MSC class 1 is mainly expressed by all nucleated cells, right? The MSC class 1 are mainly expressed by the nucleated cells. So you take any nucleated cells, they have a capacity to express the MSC class 1 and they can present those antigens, right? So if you see this MSC molecule, the MSC class 1 molecule, it's made up of two molecules, the domains, the dimer of alpha and beta. So alpha is a 45 kilodalton, whereas the beta is kind of 12 kilodalton molecule. And alpha chain again is made up of three domains, alpha 1, alpha 2, and alpha 3, almost comprising 90 amino acids, right? And apart from these two chains, the alpha chain is secured safely into the transmembrane, the membrane of a cell, to a transmembrane domain, mainly made up of 25 hydrophobic amino acids. And also the short cytoplasmic tail made up of 30 hydrophilic as well as charged amino acids. So this alpha 1 and alpha 2 peptides, the domain alpha 1 and alpha 2 domain, it makes a groove or it makes a cleft wherein the antigenic peptide will bind. And these antigenic cleft, it has the capacity to bind only 8 to 10 amino acids, and more than that it cannot accommodate. Okay, so among these peptides, if the peptides, the amino acids like glycine, proline, leucine, they occupy at second, third, and ninth position respectively. So if you see the MSC class 1 molecule, the class 2 molecules are like they contain entirely two different chains. Whereas in class 1, it is a alpha 1, alpha 2, alpha 3, a very big alpha chain and a very small beta 2 microtubule chain. Whereas in MSC2 molecule, the two chains are separate and they are made up of alpha 1 and alpha 2 chain, whereas the beta chain is made up of beta 1 and beta 2 domain. So the alpha chain is composed of alpha 1, alpha 2 as explained, and beta 2, and transmembrane almost they are very similar to the MSC class 1 molecule. Again, they are made up of 25 amino acids, and also the cytoplasmic tail, which are made up of charged amino acids. So here the alpha 1 and the alpha 1 and beta 2, again they form a cleft for binding of those antigens, the peptides derived from antigens. And the alpha 2 and the beta 2 can interact with CD4 of the Th cells. So usually the peptide cleft that is found between alpha 1 and alpha beta 1, they can hold more than 8 to 10 amino acids, particularly like the amino acid. If the peptide contains 13 to 18 amino acids, they are more highly preferred for this MHC class 2 molecules. And the anchor molecule in the sense, they need to be present. So then only they can bind to this peptide cleft. So the anchor molecule amino acids are like tyrosine and isoleucine at second and ninth position respectively. So once we see that how the structure of MSC class 1 and class 2 molecules, so I just wanted to give a detailed, I mean not detailed, like a very simplified way that how these MSC class 1 and class 2 molecule, they present those antigens on different cells. So consider this is the cell that need to be detected by cytotoxic cells, and imagine the pathogen if it enters into the cytosol or enters into the cell, and then the cellular machinery. Basically, this is the nucleated cells which is going to present MSC class 1 molecules. So once the antigen enters into the system, they are recognized by the cell proteasomal system. So the proteasomal system will degrade those antigens into smaller pieces. And then these smaller pieces are pushed inside the endoplasmic reticulum. So meanwhile, the cell gets the signal for the production of or the synthesis of MHC molecule, that is MHC alpha chain as well as beta 2 chain. And here the MSC alpha 2 chain, I mean the MSC alpha chain, as and when we synthesize, it undergo folding with the help of calnexin as well as calreticulin molecule, so that the proper folding will occur within the lumen of the endoplasmic reticulum. Okay, in fact, the rough endoplasmic reticulum. And then once the proper folding has occurred, the beta 2 microglobulin will bind to the alpha 3 domain of MSC class 1 alpha chain, and then the ERP57, so that makes the way to bind to this MSC1. So once the ERP57 binds with calreticulin, so that will drag the MSC1 molecule towards the TAP molecule. So these TAP molecules are ATP-driven molecules, so that they need energy in the form of ATP hydrolysis. So once the peptide they bind and also the ATP hydrolysis, all these peptides are pushed inside the endoplasmic reticulum, so that they can come in contact with these MSC molecules. And once the MSC molecules are completely synthesized, then MSC molecules are taken towards these TAP molecules with the help of a very special molecule called tapacin. So tapacin has got affinity for both TAP as well as the MSC molecule. So these tapacin, they drag these molecules, MSC molecules, which are unbound with the peptide. And when they see the peptide, they bind to these peptides. And then vesicles are formed, and then these vesicles, they fuse with Golgi network so that they can make a post-translational modification. And then the vesicles, probably the COP1 and the COP2, I will not go in detail about those vesicles. So maybe through a COP1 and COP2 vesicles, so they are presented on the cell surface. So this is the nucleated cells. So all kind of nucleated cells, they can produce these, they can express these MSC class 1 molecule along with the peptides that are derived from the pathogen that have been entered into the nucleated cells. So now the MSC class 1 molecules are recognized by cytotoxic T cells. So that once they bind with the help of TCR, that is T cell receptors, these cytotoxic cells, they get activated, and then they are differentiated into memory cells as well as the CTLS. And the CTLS, they can come back and they can kill these infected cell, or they can also release different kind of, what you can say, ligands, death ligands, and then they can take over those ligands. Okay, so let me finish up this quickly. So another thing, another pathway that is antigen is using the MSC class 2 molecules. The class 2 molecules are presented on the cell surface by the endosomal or lysosomal degradation. Once the antigen that is present on the cell surface, they are taken by these cells by different means, like I can say like a receptor-mediated endocytosis, or it could be the phagocytosis, or it could be the pinocytosis. So the antigens, they are taken inside, and then they are through, of course, through a clathrin-coated vesicles, and these vesicles, they fuse with the lysosome molecules, and they are termed as early endosomes, because the lysosome, as we know, they contain very low pH. So as soon as they come in contact with the low pH, so the antigen and antibody, they get separated, and then those peptides are chopped into small pieces, and then they are expressed on the MSC class 2 molecule. So here the MSC class 2 molecules, once they are synthesized from the endoplasmic reticulum and post-translationally modified, they can bind with a very important molecule called invariant chain, so that they can preserve this three-dimensional structure. And then the same complex is again, they fuse with the lysosomal degradation, so that the invariant chain is cleaved into small pieces. So only what is left is the clip. So the clip is again replaced with HLA-DM or HLA-DO, so that the peptides that are generated during the endosomal degradation, they come and they bind and they replace with this peptide, and then MSC2 molecules are expressed on the all kind of professional antigen presenting cells. So now these MSC2 molecules with the peptides are recognized by mainly the TH cells. So the TH cells will bind with the help of CD4 molecules, and then they can execute the different immune response. So to summarize the talk, what I would like to say that the MSC molecule, right? Let me go back to the same slide. So the MSC molecule, they play a very, very important role in executing either humoral immunity or cell-mediated immunity or NK cell. On the whole, the MSC molecule are the central regulators of the adaptive immunity. Thank you. So, yeah, I think you can, if you have any doubts, I would like to answer those doubts. Pramod?
Hello, sir.
Yeah, can you hear me?
Yes, sir. Yes, sir.
Okay, okay, yeah. Thank you.
Thank you. Thank you very much for your wonderful presentation and informative. It's very informative, sir. Mechanism of adaptive and innate immunity, different mechanism and antibodies, phagocytes, T4 cells, and infection process of MSC molecules. Wonderful presentation, sir. Thank you, sir.
Thank you, Pramod. Thank you, Pramod, and thank you, Santosh. Thank you very much.
So now it's for discussion.
Okay, okay, yeah, that's fine.
So we have received questions from our participants, various
Okay.
participants. So I will ask only a few questions of common,
Sure.
which are common. So first one is, can you explain the role of clonal selections in immune response?
Clonal selection.
In immune response.
Hello.
Hello, sir.
Hello.
Clonal selection in immune response.
Clonal selection in immune response.
Yes, sir. Yes, sir.
So what exactly the question? I need to explain the clonal selection.
Role of clonal selection in immune response.
Okay. Hello.
Sir.
Can you hear me?
Yes, sir. Yes, sir. You can continue, sir.
Hello.
Sir. You can continue, sir.
I can continue? Okay. 9 case okay.
Okay. So one more question, sir.
Yeah.
Is there any method available for detection of COVID based on antibody detection?
Hello. Yeah.
Hello, sir. Is
Hello.
there any one more question, sir?
Yeah.
Is there any method available based on antibody antigen detection for COVID-19?
I think already the system has developed. I mean, the many people have developed those kits, right?
Okay, sir.
Particularly targeting the envelope protein.
Okay.
So whatever the antigen antibodies, so they need to be the protein, right?
Okay.
So I think already the many people, many companies have they produce the kit mainly based on the antigen antibody stuff, I guess.
Okay, sir.
Yeah.
Thank you, sir. Thank you for your dedicated time for our session.
Oh, it's my
So,
pleasure. Okay.
on
Thank
behalf
you so
of
much.
Department of Biochemistry, Davangere University, Davangere, and Society of Biological Chemists, I heartily thank you, sir. Thank you for your busy schedule. So it's a wonderful session for especially for the students. It's a very informative session, sir. Thank you, sir. Thank you
Yeah,
very much.
particularly I concentrate only for students, so I didn't mean to tell any the experts. Anyway, that's why.
Okay, sir. Thank you, sir.
Yeah, thank you. Thank you for providing me an opportunity, and also I thank the organizers as well as Society for Biological Chemists. And thank you so much. Thank you. Thank you.
Thank you. Thank you, sir. So, so thank you all participants. So we have completed session first. So just for 5 minutes, within 5 minutes, we can join for session two. Presenting Dr. Praveen Kumar SK. So please be online. So Vishwanath sir.
Yeah, I can stop sharing. Right?
Yeah, so please stop your sharings. Thank you, sir.
Yes, thank you.
Hello, everyone. Let's we start second session two. So it's my privilege to introduce one of the speaker, Dr. Praveen Kumar SK. He's working as assistant professor, uh, Department of Biochemistry, Department of Biochemistry, Karnatak University, Karnatak University, Dharwad. So Praveen Kumar, Dr. Praveen Kumar completed MSc and obtained MSc and PhD degree from Gulbarga University, Gulbarga. After completion of degree, he joined as a postdoctoral fellow, Department of Biochemistry, Indian Institute of Science, Bangalore. Sir has received many grants and awards. Sir has received the Senior Research Fellowship, CSIR, SRF, Council for Scientific and Industrial Research, New Delhi, and Sir awarded Research Associate, Department of Biotechnology, Government of India, and Sir has received grant from UGC, startup grant from UGC New Delhi, and Praveen Sir has published more than 10 research papers in national and international journals, and he awarded best poster presentations, and best oral presentation in national seminars. So now I request Dr. Praveen Kumar SK, you begin, start the sessions to start the session.
Oh,
Praveen Kumar.
yeah, yeah. Very good afternoon, Santosh. So it's a very privilege for me to be a part of this webinar series, and I would like to first of all thank all the organizers for uh, inviting me to present or to share like my views regarding a small piece of work. Like as the morning session has gone, it was more of a theory rather than like a uh, practical session. So henceforth, even my presentation would be like more of the theory aspects which have been covered, and henceforth, like I would just begin the thing before uh, initiating my presentation. First, like I would like to thank all the organizers once again, and then and the friends of mine, Dr. Santosh, to kind enough to invite me for this session, and Dr. Pramod, Dr. Kumar sir, and Dr. Gopal Advera sir. So with these things, now let me get into the presentation. And the title of the presentation is like experimental methods in the immunology. So as we are aware, so like the life is just, I'll give you a brief idea, like the brief background of it, wherein which, so here it is something like, okay, I'll just start with a brief background, so wherein which, yeah, so as we all are aware, the term immunology, so what do you mean by like the term immunology, which has been coined from the Latin term, so it means exempt. So the definition of the immunology is something like it's an integrated body system of an organ or the tissues and the cells and the cell products that differentiate self from the non-self and neutralize potentially pathogenic organisms. The body defends against the various, like what do you call, uh, the disease-causing organisms, malfunctioning cells, and the foreign particles. So here, this immunologist, majorly the immunologists, they use the tool, like majorly from the structural biologists or the biochemistry and the cell biologist, anatomologist, microbiologist, and various physicists. And then with the brief idea what the concrete is being done with all these biologists, they come to a conclusion of how to generate the antibodies, and then how these generated antibodies, they can be used in analyzing the antigen antibody interaction. So majorly my study would be majorly concentrating on a kind of antigen and antibody interactions, wherein which the antigens are something like a, so as it would be a very simple definition as we all are aware, that is antigen. Antigen is something like it's a molecule or a pathogen capable of elicting an immune response in that particular molecule or a pathogen is specifically called as an antigen. I think hope my voice is clearly audible to all of you.
Yes, yes, yes. You can
Yeah,
continue.
that's fine. Yeah, and an antigen maybe either it can be anything. It could be a virus, it could be a bacteria, or it could be anything, and it could be any a protein or any other macromolecules. So this antigen could be like uh, these are the one which could elect or which could initiate the immune response of the body, and they have a specific markers located on a surface of these, and that is called as like a epitopes. So here, like just a brief regarding, and these epitopes are like the functions of these antigens are they are they induce the immune responses, and very important thing to know about is they are very specific. They are very specific in binding with the specific antibodies. Based on the functions, an antigen is classified into either it could be an complete antigen or a partial antigen. So the antigens may be of two types, as I said, complete or a partial antigen. The complete antigen is this antigen is having a capacity to induce, okay, antibody as a whole. But in case of the partial antigen, it is the non-specific or a specific non-protein substance is capable of inducing. So here it's a hapten molecule which is responsible for inducing or initiating the immune response. So that kind of particles can be called as a partial antigen. So this one brief idea about an antigen. Then coming to the next one, it is the antibodies. So these antibodies are nothing but it's an immunoglobulins, or basically they are the glycoprotein molecule formed from the immune response to an antigenic stimulus. So without the antigenic stimulus, these antibodies cannot be synthesized or produced. And these antibody are synthesized or throughout the body secretions or the mucus. And these bind specific, okay, they bind the specific antigens, which in turn are activating these particular antibodies. And these gamma globulin constitute of 20 to 25% of total serum protein. So these secretes, or these are visually secreted by the B cells, and these antibodies, they have, as we have seen, the structures of these antibodies. They have the heavy chains, they have a light chain, and then this is a specific site at which all this antigen, or this is called as antigen binding site. They have the light chain, they have this heavy chain as well, and this is what a basic skeletal structure of any immunoglobulins. Okay, and again, based upon their physicochemical differences, they are again of almost five types. As you can see it on the presentation, they could be IgG, there could be IgA, it could be IgM, and it could be IgD, followed by the IgE. And these immunoglobulins, they have a specific function, or they have their own a specific role to be played as a whole. Okay, so as we can see, many of them are monomers, and few of them they are like the dimers or the pentamers. Basically, the IgA work in a dimer conditions, whereas IgM plays a situations in the pentameric form. Okay, so this was a brief idea regarding the antigen and antibody. So now coming to an important discussion is like these antigen antibody reactions are basically because of the humoral immunity, or it's basically because of the humoral immunity, wherein which you could see that the humoral immunity relies on the action of antibodies circulating through the body. So here it is something like, see, when we see this small cartoon wherein which we could see that this is what the antigen or the bacterium, wherein which when it comes and bind with a B cell receptors, so these in turn induces, okay, these in turn induces the CD, okay, this in turn induces class of reactions or series of cascade of reactions, wherein which MHC2, as Professor Vishwanath was talking about MSC1 and MSC2, and here the MSC2, this induces the production of the two kinds of cells, which is called as the memory cells, and the other one is the plasma cells. And these memory cells, they play a role in the future infections, whereas the plasma cell, they counteract, they act at current infections or the current scenario. So this is how these antigen antibody interactions are being triggered in a immune system or in the body. So here each antibody bind, okay, so here, so as we have seen, so these antigen antibody interactions are more or less something like, as we have seen in case of the enzymology, wherein which we could study a very beautiful hypothesis, wherein which they say that like lock and key hypothesis, the similar hypothesis do play a role in having an antigen interacting with a specific uh, like a motif, wherein which it binds to the antibody. And these are single reactions which is being or complex which is being formed is called as a singular immune complex. And these, the moment they form a kind of a immune complexes, they tend to trigger a series of reactions such as inflammation, as we can see that complement activation, opsonization of a target, and then that is how the series of things initiates, and then they would destroy the pathogenic compound. So as we all are like kind of a biochemist, so we need to know about what are the kind of interactions that are being happening. So as in this particular picture, you can see that there are various types of bonds which have been formed between the antigen and the antibody. Earlier, what we thought was like something like it's a simple like antigen antibody interaction. But if you just look at this particular picture, you could see is how or what are the different types of bond that are being playing a role in having an interaction with antigen with our antibody, wherein which we could see like there are like hydrogen bonds are being formed. You could see that there's like formation of a hydrogen bond form, then ionic ionic bonds, hydrophobic interactions, van der Waals interactions. Okay, and along with that, you could see the ionic bond. These are some of the various important bonds that are being formed. Okay, and with help of which a firm binding or a kind of a, it's a non-covalent bonding, which is, we could see that there are different types of bonds. So this is a totally a combination of several bonds, but it is a non-covalent interaction that is being formed between like this antigen present and then the antibody structure. Okay, so these are some of the important pictures which I have taken it from textbooks, wherein which this portion is antigen, wherein which it very closely fit into the structure of the antibody. So both of them, they have a very like how we have seen it in case of the lock and key models. Okay, so now we will just venture into the next level of discussion, that is how is antibodies are generation, and then in the very important part or the main theme of my talk would be like the experimental part, and that experimental part I will just get into the next few minutes. Okay, so here it is something like, so again the antibodies which have been generated by a simple immunization method. So here by simple immunization method, so we could get two types of like what do you call the antibodies. One would be like monoclonal antibodies and then the polyclonal antibodies. So we people are very quite familiar with these kind of the terms, that is monoclonal and the polyclonal, where in which see, these type of antibodies have been produced, and these polyclonal antibody they have some disadvantages, as we all are aware. Polyclonal means so it can bind to a multiple epitopes, whereas the monoclonal is going to be very specific. To avoid the cross reactivity or the problem or the disadvantages of these polyclonal antibodies, so the monoclonal antibody productions is going to play a very important role. So here, in year way back in 1975, okay, George Kohler and Caesar Milston, they figured out how to generate these uh, monoclonal antibody using mice or the hybridoma cells. And these are some of the cancerous cells. So here, this image or this is a flow diagram in which we could see that this is an like an antigen. This is an antigen wherein which you could see that there are four specific epitopes, and this antigen is now introduced into the mice and then followed by the different steps, that is they isolate the specific polyclonal. So this serum which is being isolated after introduction of this epitope or antigen is called as the polyclonal antiserum. So how would we get into the monoclonal antibodies? So here, these isolated spleen are now allowed to react with individually the myelomas, or as we have seen, it's a cancerous cells, and this in turn this is being used. This methodology is being used for production of a monoclonal antibody, and these monoclonal antibodies are going to be very specific. The antibody which is being synthesized against the antigen 1 would be antibody 1, and so on. Okay, so here, like, yeah, now the main theme of my concept is something like immune reactions, wherein which what are the different types of immune reactions that are being carried out. Majorly, the precipitation, the agglutination, if enzyme link immunosorbent assay, or the ELISA, or chemiluminescence immunoassay, and immunofluorescence, and the flow cytometry. So in these discussions, I would like to give a brief overview of all these type of antigen antibody reactions that are being used in a kind of a diagnostic assessments. Okay, there in which these are the most prominently used methods. So let me get into the first one, which is called as immunoprecipitation. So immunoprecipitation is something like it is this. Okay, excuse me. The multivalency of an antibodies have. So here in this case, the antibody which can interact with the different antigens. Okay, and this technique is being used for studying, and this is what one of the most earlier concepts which was developed. Okay, so and this is something like they are quite vulnerable, and but still there are many tests which has been based upon these immuno precipitation concepts. So here immunoprecipitation can be carried out in case of the solution as well as in the gel, as we have seen. So in case of the solution, it's something like when an antibody and a soluble antigen are mixed in a solution, the bi or a multivalent nature of this immunoglobulin, this allows for a single antibody molecule to bind to form an, okay, so they form a complex ligand. Okay, so as I'll just give you this. So here to form a antigen antibody complex with a lattice. So here the amount of this graph gives a clear picture wherein which if at all the antibodies are less in number or the antigen are less in number, this cannot form a proper cross-linked precipitate. For the formation of the precipitate, we require equivalent amount of antigen or antibody, as we have studied in case of the enzymology, wherein which if at all the substrate is falling short or the enzymes are falling short, it cannot give a 100%, or the amount of substrate conversion to the product is limited. Same way, the same concept is working here as well, wherein which, so if you see, look at this axis, which is x-axis, wherein which the antigen is going to be added. And in this case, the y-axis, you could see that the antibody precipitation rate can be assessed. So here, this is called as the pro zone, wherein which if we go on, you could see at the upper part of the screen, wherein which there are plenty of freely available antibodies. Okay, wherein which there are only few antigens, but this could not form a cross-linked precipitate. Okay, and if you get into the next level as well, wherein which antigen are in excess, even that they're not going to lead formation. So here we need the equivalent, or it's called as the equivalent zone, and at that particular zone we could see that the precipitate of this is being formed. Okay, this is all about the solution, wherein which this is like any general chemical precipitation test that we could do it in case of the test tube. So now coming to the immunoprecipitation gel, or in case of the gel matrices, immunoprecipitation in case of the gel matrices, wherein which you could see that. So probably we all have seen the best example would be like the Australoni methods, wherein which, so here we are preferably we're going to use the agarose matrix. Okay, and then antigen and antibody diffuse together to one another, towards one another, and then they form a visible line of precipitation will be formed, and that is what which indicate the specificity of the antigen and the antibody. So So in this method is something like it can be very clearly visible. Okay, so if you look at this particular picture, so wherein which, so this is an example wherein which an antiviral substance is added at the center well. Okay, and then anti sera has been taken from the different individuals, and then they are being placed at A, B, C, D, and we could see that a thin line formation, precipitation line formation can be seen at all the wells except for the two wells, which is B and D. So this indicates that B and D are giving a negative results, or this doesn't have any antiviral antibodies. So henceforth, this is not okay. So these kind of assays are being used in case of the immunoprecipitations. Okay, then coming to the next setup, wherein which we could see the agglutinations. So agglutination reaction is something like, so in case of here, so here the antigen was soluble in case of the precipitate, but whereas in case of the agglutination, it is the insoluble one. So agglutination technique is like it's a more advanced, or even say a more sensitive one, rather than when we get back to the earlier precipitation one. So it's going to be the more sensitive compared to that of the earlier precipitation reaction. So here, the clumps reaction is called as agglutination, and the causative agent which causes this clumping is called as the agglutinin. So here that we need to note it down. Yeah, the agglutination reactions are identical as, and this we could see. So this is the best example that I have taken it here for to understand the concepts. So whenever probably like the blood group detection, probably everyone might have done this particular experiment in your laboratories to confirm the blood groups. So that would be one of the best examples. And here I've taken a different one wherein which we are using the hemoglobin. So whenever we are using the hemoglobin, and that particular reaction is called as the heme agglutinations. So here, so this is something like a microtiter wherein which the RBC is allowed to interact with different viruses and the different antibodies. There are the three different, what do you call, the cases. In the first case, it's a purely RBC, which has been allowed in the microtiter plate, and you could see there is no agglutination formation or no clumping is being occurred. But once when in case of microtiter plate, wherein which the virus is being added along with the RBCs, we could see that there is a formation of a clumps, or the agglutination has been occurred. And in the next case, wherein which the C stage, the virus plus antibody plus RBC, these are the three combinations wherein which what happened. The viruses which were free to interact with the RBCs in the B case is not available, because those are now bound by the antibodies. It's a kind of a hemagglutination inhibition with the test which have been done in the regular one. And we could see that there is no clumping formation in case of C as well as the A. Only the clumping is been occurred. So these kind of studies are called as the heme agglutination reactions. Now coming to a very important kind of analysis, or the assay methods, which is very widely used. It is something like ELISA. Probably everyone is aware of these things. It is as abbreviated. It is enzyme-linked immunosorbent assay. Wherein with, so here, so this method is very sensitive method. So this is very widely used in many clinical uh, analysis and diagnostics. So, so this in this case, so as I have not included radio immunoassay, which is again one of the uh, commonly used technique, but as it is outdated. So nowadays, none of the chemical or none of the diagnostic or the research people, they do promote this use of this radio immunoassay. Henceforth, I have skipped it. So here I have now just ventured into the next one, that is the ELISA, wherein which here again, as the name starts with enzyme linked, that means the enzyme is conjugated, and then so this is now conjugation of enzyme that plays a basic for their ability to catalyze or convert the substrate into colored fluorescence. And most commonly used enzymes are like orthosteric peroxidase, alkaline phosphatase, and glucose oxidase. This is going to be like more sensitive, and it's having several advantages over the radioimmunoassay, which is going to be like a biohazardous. And there again, a very this ELISA has now been developed into different formats, such as and this ELISA method can be used for quantification as well, quantification of the antigen or the antibody as well. So these are some of the different types of ELISA techniques, that is direct, indirect, sandwich ELISA, and then finally the competitive ELISA. And as you could see, I'll just give an overview of all these types. So in case of the direct ELISA, wherein which this is for the microtiter plate, wherein which the antigen is coated onto it. And then to that, we going to add the conjugated antibody to that particular well, and then finally wash it, then look it, observe for, look it. So, so here what's going to happen is whatever the substrate which is being now converted into the colored one, that can be measured using the ELISA readers, which are being available in different. So we have a spectrophotometric, colorimetric, and then different types of readers are available in uh, our laboratories, or which can be used. So the next type of ELISA method is the indirect one. So in this indirect session, it is sorry, indirect one. It is antigen which has been coded over here. So that we have here in this case we require two antibodies. One is the uh, primary antibody. Sorry, yeah. So here it is like the primary antibody, and then to that you have a conjugated secondary antibody which comes into picture, and then this secondary antibody which is being now linked with the enzyme, and that in turn converts the substrate into the colored product. Okay, next coming to the next type, wherein which it's the sandwich ELISA method. So in case of the sandwich ELISA method, so here in whatever the other previous two method that we have discussed, the well is being coated with antigen, but in this case it's going to be like the coated with the antibodies. So here the primary antibodies are now coated onto the wells, and to that antigen is being added. To that the secondary antigen, which is sorry antibody, which is coated with enzymes, are loaded onto it, and that in turn converts this or this reaction it initiates, and this gives a colored reactions, and this can be read by adding plate reader concepts. Okay, then next is the competitive ELISA, wherein which there is it's you could see that there are two types of antibodies. One is which is inhibitor for these particular antibodies. So here a competitor is being incorporated in this particular ELISA method, wherein which they are coated, and then here only the specific antibody is bound to the antigen specifically, but the competitor is not been bound. So here the secondary antibody will binds to this one, and then that in turn converts this substrates into the product, and then the product form is red. And the next important method that's been used is the chemiluminescence, which is gaining a like nowadays, which is in a boom, as we could see in case of the research. So we have like many people who are into active researcher, they are now into chemiluminescence. Instead of a gel dock, they go for a chemiluminescence docking and all. So here in which here again, this is a kind of a a kind of advanced technology wherein which they use the radio. In this method, they use a different, we call the or the chemical reaction. So in this phenomena, it is something like emission of energy in a form of light in some chemical reactions. That means a kind of a dye or a kind of a substrate which is being used, and that in turn, with the help of an enzymatic conversions, which in turn triggers the light or which generate the light signal. This kind of studies are being used for, or in case of the chemiluminescence assay. So here it is like, uh, okay, we could see that this is what the HRP, that is a hot reduced peroxidase, this is an enzyme, and this enzyme which in turn converts into the chemiluminescence substrate into the light source, and this could be used. Okay, so here it is the compound luminol. Okay, that is the H2O2, and then this orthodox peroxidase which converts, it produces the light. Okay, and now let me, yeah, move it quicker. So we all are aware of microscopic visualization of the cells. So again, here also we could use um, so in a early steps, it is something like we would have gone for a basic, what do you call it, the bright field microscopes or the dark field microscopes, or the phase contrast one, wherein which it's something like we are going to, or the histochemical or the histological studies, what we are able to see in case of the zoology laboratories and all, wherein which the cells are being now fixed, and to that cells, they are being flooded with a different colored dyes. Okay, and this could be seen, visualized by using a specific, what do you call, yeah, you need not to go for any higher level of uh, instrumentation as well. But when it comes to the immunofluorescence, so this is something like a basic one, wherein which you could use a different dyes. Okay, then comes the important one is the immunofluorescence based immune technique, wherein which here I think we people are very well versed with the fluorescence. What is this fluorescence? So the phenomena of fluorescence result from the property of any molecule which can absorb the light of one wavelength, and in turn it get excited, and then it emits the wavelength at a longer longer wavelength. That means if at all the emitted wavelength is a longer wavelength, and it falls in the visible spectrum, that kind of things, that kind of compounds are the compounds are being very useful in this kind of technique. So here, or else, see, nowadays there are many uh, engineered proteins or engineered proteins which have been there. Probably everyone might be aware of the green fluorescent protein or the red fluorescent protein. These are again a proteins which in turn get excited about hitting with a specific light. Okay, and along with that, there are different dyes as well which are there in the market, which could be used to bind specifically to specific biomolecules, such as like I think everyone might have heard about the DAPI, which is very commonly used, diamino amidino to phenylindole. So this is which has been very much used for staining of the DNA. So this is a very beautiful picture which has been taken from the immunofluorescence staining of von Willebrand factors. So these are the endothelial cells, and these are because of the green color is because of the polydine, and then the blue color stains. This is because of the DAPI, which uptakes the nucleus, preferably the DNA molecule which are there, or the nucleic acids which are there in the, okay, nucleus. So this is again one more important fluorescent picture, wherein which we could see the different colors, the green color, red color, blue color, and these are all the major different pigments, majorly because of the different dyes which binds to a specific. So this is like more of a kind of a microscopic. So if you want to if you want to look for a mitochondria, you can get directly into the mitochondria, or you can strain a specific mitochondria. And if you want to strain any other biomolecule, sorry, any other subcellular portion, you can use a specific dyes which have been very commonly available in the market nowadays. Okay, so this is like immunofluorescence is not going to be like a very powerful approach nowadays. So wherein which we have a very specific and more exploited nowadays. Okay, and now our last important or last important technique which is being predominantly used nowadays is the flow cytometry. The flow cytometry is something like, it's a modern tool, so which has been used or which has been developed by Harzenbergs and the colleagues, and found some. They found the very important or the earliest application in analyzing the blood, notably the lymphocytes. So flow cytometry is something like, nowadays you cannot just think of a laboratory which is into what do you call uh, which is into major research. So you cannot expect, you cannot think the lab without these equipment. So this is going to be a very important equipment, and this is something like, it's a collective. This instrument, single instrument, work in such a large principle that it can it can what do you call it? It can replace many important equipments. So here the technique or the analytical technique that is being used is like, it can quantify the frequencies of cell bound to the fluorescent antibodies are scattering light. So it is something like, it's much more advanced technique, tools or technique, or which is being used in the fluorescence. And now there are this flow cytometry, which is now being influenced with the help of a fluorescence. Okay, so here they adopt. In this case, we could sort. That means sorting means we can divide the cells, number of cells which has been analyzed. We can divide each and individual cells into what exactly is its size, shape, what are its morphological changes, what are the internal structures, everything we could study with this flow cytometry. And whenever this flow cytometry is now what do you call, is accompanied by the fluorescence, that kind of studies is called as FACS, or fluorescence activated cell sorter. So this is again a, so same instrument which is being now kept employing the fluorescence studies. So here, this fluorocytometry uses a laser beam and a series of detectors. So here in this case, you could, if you just look at it, how the principle of this flow cytometry works is going to be like very interesting, wherein which you could see each and a very narrow, with a very narrow tube, the cells, if you just see this image, you could see all the cells are now stained with the different colors. You can see a very narrow tubing with which the cell are now passed. This is the laser beam which is now allowed to pass through that, and each, so there are many detectors. So whatever the, if you see the spectrophotometer, we have only, we could see that yes, we could scan only the optical density, or we could scan the transmittance. But here, this flow cytometry is a kind of a collective of multiple equipments which are fused together. So here you can quantify the fluorescence, you can quantify the what do you call, the light which is being refracted, the light which is reflected, many things you can do it in a single most or single equipment. So here, so if you just look at this, this is what the sample preparation, you could see there are multiple cells, green cells, red cells, the gray, the white one, everyone. So here it is being now allowed through a hydrodynamic, that means a complete in a liquid sheet fluid, it is allowed to pass, and then each and every individual cells. So in a minute, it can sort like there are several thousands of the cells, and then you could see that there are series of detectors there. See, you could see the side scatter, the forward scatter, and these are the detectors which are located on different angles. So these are specifically for the fluorescence. Again, there are some. So these are the dichroic mirrors. So probably while discussing about this, what do you call the fluorescence microscopy, we could have come across these particular mirrors, that is dichroic, which allow only specific type of light. So it is something like it will act as an opaque, as well as it is allowing only one type of or one wavelength is being allowed at a that particular part of it. So this is something like this is what the instrument setup. So with which we could screen, uh, which is being used for analyzing the various uh, cells, cells physiological studies, cell what do you call the condition, physiological condition, whether it's healthy cell or it's unhealthy, everything can be sorted out using this particular study. So this is like an overview, or this is like a what do you call one dimension result that we could get it after when we run the flow cytometry. So it is like the forward scattering. So if we just look at this results is going to be like a interesting one. So here these portion, which is like what do you call, these are the lymphocytes. These are going to be like very small cells which are on this portion, and then these are the monocytes which are a bit larger than the lymphocyte. They are now sorted in a different locations, and then you have neutrophils again which are being large enough cells. So henceforth these flow cytometry methods are going to be very is like very widely used technique nowadays for analysis of this. So here the laser beam and the detectors which have been placed that are going to play an important role in the feed fluid, which is being like which maintains the hydrodynamicity of the flow. Okay, so probably so this technology, okay, so this can be capable of detecting a, so based upon the detectors, so it can detect up to 8 to 12 fluorescence, and the light scattered parameters are very commonly used nowadays in a research or the regular laboratories. Okay, so with this, I think hopefully I could give you a brief overview of how the antigen antibody interactions occur in case of the precipitation agglutination method, and then followed by ELISA method for, and then chemiluminescence, immunofluorescence, and then immuno. Okay. So with this, probably like I would like to wind up the discussions what I have done. Yeah. Hello.
Thank you, sir. Thank you, Praveen, sir.
Yeah. Yeah. Thank you, Santosh. Thanks a lot.
Thank you. Thank you very much for wonderful presentation and very good information in this session, especially basic immunology related to antigen antibody interaction structure and different immunochemical techniques for detection of diseases. And it's very informative. And participants have raised some questions. So
Okay. Yeah. Yeah.
discuss the questions. So we
Yeah.
have
Yeah.
selected few questions which are common. So
Yeah.
how monoclonal antibodies can be used to treat certain types of cancers? First question.
Okay. So again, see, as we have seen, the advantage of monoclonal antibody is something like they act on a specific epitope.
No.
Okay, and then if at all we could see what is the what is exactly the what do you call the if you could make the study of this epitope, then monoclonal antibody would be very uh, advantageous for treating the cancers cell.
Oh. Okay, sir. One more question.
Yeah, yeah.
Among the all immunological reactions, which one
Okay.
is most widely used, or which one is effective.
Okay, effective in the sense like whatever like nowadays the ELISA method or the immuno precipitation method, which have been very commonly used for any diagnostics and all. That would be the most effective one. What I feel is that. I,
Okay, sir. Uh, Thank you, sir.
okay.
On behalf of Department of Biochemistry, Davangere University, Davangere, and
Mhm.
Society of Physical Chemists, I
Okay.
thank you once again. Thanks for participation and thanks for sharing your information. Thank you,
Okay.
sir.
Yeah, I would like to. I would like to thank all of the organizing team once again. Thank you me giving me an opportunity to share my viz on it. So uh, finally, like I would like to thank one and all the participants for their patient hearing. Thank you.
Thank you, sir. So
Okay.
can you please unshare the slides?
Yep. I'll just stop sharing. Yeah.
So all participants, a small notice for all the participants. Uh, Tomorrow we have two sessions. Two sessions due to unavoidable circumstances and unavoidable conditions. Dr. Divya Prakash is not, Madam is not available tomorrow. Due to this, in continuation of this, we have one more eminent speaker, Dr. Nagaraj, Assistant Professor, Tumkur University, Tumkur, on experimental animal models in biochemical research. So tomorrow will be two sessions. The one session is replaced by Dr. Nagaraju, Assistant Professor, on experimental animal models in biochemical reactions. So thank you all the participants for participating the fifth day session. Thank you one and all.